I'm Daniel Levine, and this is the Bio Report. One of the promises of AI is to redefine what's possible by enabling the discovery of compounds that exist in a much larger chemical space than scientists have previously been able to explore. Deepcure is using AI and physics to discover small molecule therapies that can bind to difficult to target proteins. We spoke to Kfir Schreiber, co-founder and CEO of Deepcure, about the company's AI technology platform, its focus on autoimmune diseases, and why he believes its technology will allow it to develop small molecule drugs that can provide alternatives to biologics to treat these conditions. Kfir, thanks for joining us.
Thank you, Danny. Happy to be here.
We're going to talk about immune diseases, Deepcure, and its AI platform for discovering new small molecule therapies to treat these conditions. Let's start with the challenges the time and cost of drug discovery and development pose. We've seen a steady stream of innovative technologies to change this, yet they haven't seemed to move the needle. Why does it remain so expensive?
Yeah, Danny, that's a great question. And I think the first thing we need to understand is that drug discovery and development is this extremely complex multifactorial problem with many different stages. So you can think about it anywhere from basic science, the discovery of a new therapeutic agent, then preclinical development, and of course, clinical development. And when you look at the relative cost of each of these stages, it's pretty obvious that the discovery phase is not really the most expensive one, at least not in out-of-pocket dollars. And when we think about what Deepcure is doing, which is developing novel technologies for drug discovery, we mostly focus on what kind of unsolved problems we can solve using our technology. So turning the impossible possible and not necessarily making it cheaper or faster per se.
There is a lot of excitement over the potential for applying AI in general to the problem. What's the case for why this technology might be different than other technologies we've seen come into the discovery and development process?
Yeah, so I actually don't think it's that different. I think a lot of technologies in the drug discovery space go through the same cycle of boom and bust. We've seen that after the first human genome. We've seen that with DNA-encoded libraries. We've actually seen that with machine learning and AI drug discovery. If you think about it, it's not a new concept. People have been applying machine learning technologies for drug discovery for multiple decades. But I believe that we are at this pivotal moment where we see these technologies start to deliver. We see the first real drug candidates going to the clinic. We see the first successes where some of these problems were much more challenging. I don't want to say impossible, but they were definitely much more challenging before. And now we see these technologies mature and get to a point where we can see the deliverables, we can see the output.
In the case of Deepcure's technology, is there any way to quantify any time or cost saving it's been able to achieve to date? And where specifically might it help accelerate or compress the time?
Yeah, so as I mentioned, for us, it's much more about taking fundamentally very challenging problems and solving them. So it's a binary 0 to 1 rather than doing it faster. For example, our lead program is a BRD4 BD2 inhibitor. And people have been studying this class of therapeutic targets for more than 15 years now. We've seen more than 100 clinical trials with BRD4 inhibitors. Unfortunately, all of those inhibitors were non-selective, meaning that they were also hitting and inhibiting other BRD family members. And that caused significant toxicities in the clinic, primarily thrombocytopenia. And that's why all of these molecules failed. And for that reason, we have used our technology to discover the first ever truly selective BRD4/BD2 compound. And this means that we can now mitigate and potentially completely eliminate those toxicities that people observed. And we can progress this compound now to clinical trials in chronic indications like rheumatoid arthritis and hidradenitis suppurativa with a much, much cleaner profile. So I can't necessarily say that our discovery process was X dollars cheaper, but I can say that we have done something that many, many people tried to do in the past. All of them failed, and Deepcure managed to deliver on that. And that's true for all of our discovery programs. We try to focus on extremely challenging chemistry problems that are challenging across the industry and solve those. So it's pretty hard to compare what's the efficiency gain in something that was really never done before.
It strikes me what your technology is really enabling is the exploration of a much larger chemical universe than has been traditionally possible using existing libraries. Is that a fair statement?
Yes, absolutely. And the way I think I would describe it is that we focus on mitigating design biases in the exploration of chemical space. Right? Chemical space is available to everyone, but there are multiple biases that exist in traditional drug discovery that limit the effective scope that experts in the field can utilize in their own discovery programs. And what we have done is to create a very holistic suite of tools that allow us to mitigate those biases. And let me offer a few examples to explain. The first step in every discovery program for us is to explore the protein of interest. Traditionally, drug discovery people would take either the native ligand or potentially a hit from some sort of screen, and they would use that as their binding mode. That's the place, the area of the protein they will utilize to modulate the function. Instead of doing that, what Deepcure is doing is using a combination of physics-based simulations together with artificial intelligence to identify all of the possible ways a small molecule can bind and interact with this protein, which means that we identify significantly more opportunities and we don't bias ourselves to one particular binding mode. That's just one example. We also have a generative AI tool called Morgen that is creating libraries of virtual molecules on the fly as a fit-for-purpose solution. So instead of taking a pre-existing virtual database of molecules and testing that to see if you can find a molecule of interest, we're creating a very unique library for each target we work on. Think of it as more like this algorithm you might see on your favorite video platform, where it will recommend the next series based on what you liked in the past. The same way Morgen is proposing more and more molecules based on their fit to the target of interest. So all of those are examples for how we can mitigate existing design biases in the drug discovery process to effectively increase the chemical space that is explored and evaluated to try and identify molecules for these very challenging requirements.
At some point in the discovery process, you have to move from in silico development to wet lab experiments. That means synthesizing the compounds you've discovered. How big a challenge has that been traditionally, and how does the exploration of new chemical space add to that challenge? What have you been able to do to address that?
Oh, that's a wonderful question. So when we started, we thought that, exactly like you mentioned, expanding the chemical space in silico is going to be sufficient. We can find those molecules, we can then hire a chemist in a CRO and make them. what we found out is that if you don't handle the synthesis question, you end up converging to the same type of molecules, the same type of biases other people are falling into. So basically what a lot of companies, I believe, have seen, including Deepcure, is that you can have the most sophisticated generative AI algorithm in the world that will generate the perfect drugs. But if you then need to go back to traditional manual chemistry to make them, you're going to compromise to a point where it doesn't make any sense. So to solve that, we created what we call inspired chemistry, which is a fully automated synthesis platform aimed at making drug-like molecules. A few months ago, we released a press announcement for one of the first projects for this platform, where we took an active ingredient from an approved drug, a very complex molecule for synthesis, and we synthesized that one and a few tens of other molecules around it on this automated platform. This is the first time ever such a complex molecule, 10 synthetic steps, including purifications and analytical chemistry, are being done on such a platform. And inspired chemistry has been a huge advantage to Deepcure because now we can match the state-of-the-art design capabilities we have with a synthetic capability that can make significantly more molecules that we can then put to the test in various in vitro and in vivo models that we use in our traditional screen cascade.
Autoimmune diseases have traditionally been treated with biologics rather than small molecule drugs. Why is Deepcure pursuing small molecule drugs for these indications?
Partially because they've been traditionally targeted with biologics. So when we thought about what is the ideal disease area for Deepcure, we tried to find a combination of a very significant unmet medical need. That's first and foremost. At the end, it's all about the patients and what kind of value we can deliver. But then we were also looking for an area where our technology can deliver a unique competitive advantage. And we found that inflammation and autoimmunity pose exactly these combinations. So obviously, it's a huge space with major unmet needs. But like you said, it's completely dominated by biologics and mostly antibodies. And while biologics are exciting for many reasons, they suffer from a few key limitations. They normally don't penetrate cells. They are almost never oral, so they require some form of injection, and they don't penetrate the CNS, the central nervous system. And small molecules offer exactly that. So if we can replace any antibody with a small molecule, we can replace an injection with an oral pill. We might be able to target neuroinflammation targets in the brain. We might be able to go after targets that are inside the cell. And that's exactly what we are doing. And this aligns extremely well with the very unique design capabilities that we have built. So what our platform does best is to take some of these very challenging targets, protein-protein interactions, transcription factors, extreme selectivity challenges, and design small molecules that will fit exactly that. Right? Some of these were historically considered as undruggable targets, but I think we've proven that we can turn some of these druggable. And so that's why we decided to focus on inflammation and autoimmunity.
Well, walk through the discovery process for me and the different technologies you're applying to it. Where do you start and at what point do you have a lead candidate?
Absolutely, happy to do that. And so for us, it all starts with an indication, a patient. Right? We try to find an indication and a therapeutic target that are, first of all, very well validated from a biological perspective. We specialize in designing the molecule. So we try to find therapeutic targets that are well validated, but are extremely challenging to most other people. So that's the starting point. once we identify that, and that can be BRD4, BD2, it can be STAT6, and a few other targets that we work on. Once we have that, we need some form of a structure to work with. It can be a crystal structure, it can be cryo-EM, it can be homology models, and more recently, it can be AI predicted structures. All of those can serve as a starting point for our design cycle. At that point, we apply Pocket Expander, which is the tool I mentioned before to explore the surface of the protein. Pocket Expander uses this combination of physics-based tools with AI to map the protein surface and identify all of the opportunities for the molecule to bind and modulate that protein. That sort of map is then passed to Molgen, which is our generative AI tool that creates massive libraries of molecules for that specific need. And on average, Molgen might, in silico, experiment with billions, sometimes even trillions of different design ideas to then converge to tens or potentially a few hundred of designs. And those will be then passed to our human experts. And I think that's a key point. For us, it's almost never about just the AI. It's always this back and forth between the human experts, the medicinal chemist, the structural biologist, and the AI tool. And then they go back and forth with those design ideas, providing feedback, iterating on them, tuning, and so on, until we select a handful of molecules. It can be 50, it can be 100, that are passed to our automation platform for synthesis. At that point, the molecules are being synthesized and go into the same type of wet lab experiment that any other company would use. So we start with biochemical or biophysical binding assays, we move to cellular systems, then to in vivo models, and eventually to clinical trials. So once the molecule is made, it goes through the same level of scrutiny as any other molecule. But the way it was designed is the unique piece to Deepcure.
And once you have a lead candidate, is optimization done in a traditional way?
So both the discovery of the initial lead as well as the optimization phase look pretty much the same. It's the same combination of human experts and AI tools that iterate on the either in silico de novo designs or an existing compound to try and improve the properties to fit the specific profile of interest. So in both cases, it's a very tight collaboration between the human expert and the AI physics-based platform that we have built.
And to what extent does the system learn from the wet lab experiments you conduct? Is there a way to feed back into the...
Absolutely. And I think this is potentially one of the unique pieces to Deepcure. Now, a lot of people claim that there is this feedback loop between the AI and the experimental setup. Unfortunately, if you have to rely on human chemists, whether it's internal synthesis or outsourced CRO synthesis, the turnaround time to get the molecules made, then test them, get the data, and feed it back ends up being a blocker to your ability to truly fine-tune the system. So imagine if you had a certain version of your AI model that designed 10 molecules. Now you had to go and synthesize them, test them, and 10, maybe 12 weeks later, you get data back. The AI system is no longer the same system. Your computer scientists did not sit there 3 months waiting for that data to come in. They keep iterating and innovating. So if that turnaround time is too long, the effectiveness of data coming in becomes very small. Now, what we have been able to do is using the automation platform and inspired chemistry, we can now speed up the cycle. We can get molecules made within 1 to 2 weeks. We can then test them, and then 2 or 3 weeks after the molecule was designed, we already get the data, feed it back to the algorithm, and tune it. So the shorter we can make this turnaround time, the better this cycle becomes. And I think that's a key point for the Deepcure platform.
Your company did an interesting process to validate the system. I'm wondering if you can explain what you did and what you were able to demonstrate.
Absolutely. I actually think this is probably the most exciting thing. And because, like I mentioned at the beginning, at the end, it's about the product. The AI is super exciting, but it's a tool. It's a tool that helps us deliver better drug candidates to the clinic and hopefully one day to the market and to patients. So for us, at the end of the day, it's all about the pipeline. It's the type of drug candidates we can deliver. So I mentioned, for example, our BRD4/BD2 development candidate. And this is the first time ever anyone designed a selective BRD4 inhibitor. We managed to show in multiple in vitro and in vivo systems that our drug candidate does not suffer from any of the known toxicities. I think this is a very exciting promise to patients. We now have an opportunity to take this extremely efficacious molecule and deliver it to patients in the clinic without the known toxicities. So I think that's an exciting one. And to give another example, our second program is a STAT6 inhibitor. STAT6 is a transcription factor associated with allergic diseases, asthma, atopic dermatitis. And for a long while, STAT6 was considered an undruggable target. STAT6 is a transcription factor with a PPI surface. It's really what you would consider to be impossible for a small molecule. Deepcure now has 4 different chemical series targeting STAT6 in extremely unique ways. They have really nice profiles. They are orally bioavailable. They are selective. They have very good efficacy. And we're now developing these forward. Again, this is an example of taking something that was considered impossible and turning it into a molecule at the end that one day can provide value to patients. So for me, this is the whole premise of Deepcure, and this is how we validate the approach.
Let's go back to your BD4 inhibitor. For people not familiar with BD4, what role does it play in the inflammatory process?
Yeah, so BRD4 is an epigenetic modulator. And as such, it's involved in the translation of multiple different genes. Many of them regulate inflammatory pathways. Now, historically, people developed BRD4 inhibitors for oncology rather than autoimmunity. And the reason is that those molecules had very strong anti-proliferative effect and significant toxicities. So they were not considered safe enough to be developed in autoimmune conditions that require chronic dosing and so on. And what we were able to show is that if you can design a truly selective inhibitor, most of these toxicities go away. And now we can really focus on what are the benefits of inhibiting BRD4/BD2. And what we've seen is that because of this broader effect, this multifactorial effect regulating different inflammatory pathways, BRD4 inhibitors are extremely efficacious in autoimmune conditions. We have compared DC9476, which is our development candidate, to standard of care therapies in multiple in vivo models of rheumatoid arthritis. We've used collagen-induced arthritis model, we've used a Kaya model. In all of them, our candidate completely outperforms standard of care therapies. And we believe this is extremely exciting to patients, especially patients that did not respond or stopped responding to existing therapies. And the reason we think it's a great fit for these patients is because of this multifactorial nature. If you think about it, traditional antibodies like TNF-alpha inhibitors or IL-6 inhibitors focus on one signaling pathway. And while that can be efficacious, most of these autoimmune diseases are multifactorial. They operate through multiple signaling pathways. And to be truly efficacious, we need to have an effect on most, if not all of them at the same time. So what we've been doing with our BRD4 BD2 inhibitor is exactly that. We show that we regulate multiple inflammatory pathways at the same time, almost like a combination in a pill, which ends up being extremely efficacious in various disease models.
This seems to be a huge challenge in that one of the problems with doing anything to dampen the immune system is that it could have unwanted effects. How are you able to get specificity while also being multifactorial?
Absolutely. And I think this is exactly why we got excited about this target to begin with. There was plenty of evidence before we even started working on this that the effect of BRD4 BD2, this specific domain of BRD4, is strongly associated with disease-relevant pathways, while many of the broader immunosuppressive effects were driven by either the BD1, so the first domain of BRD4, or other family members. And at this point, we have lots of preclinical data in multiple systems, in vitro and in vivo, showing that this compound does not have any suppressive effect on the hematopoietic system, right? That the most common immunosuppressive effect is a reduction in white blood cells. This compound doesn't do that. In fact, it shows recovery in disease models that mimic suppression of white blood cells. And that's exactly why we are excited about this asset and this target, because of its multifactorial but still very selective phenotype. It doesn't show any anti-proliferative effect that was seen with historical BET inhibitors. It doesn't have any thrombocytopenia associated with it. It doesn't show any suppressive effect on the hematopoietic system. So we still see very strong effect on the disease, but at this point, we don't see any of those broader immunosuppressive effects.
What additional work needs to be done preclinically, and when do you expect to advance this to the clinic?
So we are roughly halfway through our IND enabling studies. We have done most of the CMC work. We've done the majority of the non-GLP toxicology work. We still have one big toxicology study to complete before we can file the IND. And at that point, we plan to advance this to the clinic next year in 2025.
Deepcure has raised a total of $72 million to date. How far will existing capital take you?
Yeah, so I wouldn't go into specifics about the financial status of the company, but I can say that we have significant runway to continue and advance this pipeline. And our plan is to carry these compounds forward into the clinic, especially DC9476, but we're extremely excited about our STAT6 assets and others. And hopefully we want to do that in multiple indications in parallel. DC9476 will be advanced in both rheumatoid arthritis, as I mentioned, but also in Still's disease, which is a rare, mostly pediatric disease, as well as hidradenitis suppurativa, an extremely painful dermal condition. So a lot of hope for these clinical programs, and like I mentioned, we still have plenty of runway to continue and develop this.
Well, what's the plan for raising additional capital, and what's your take on the discussions you've had with investors to date?
So, I won't go into specific conversations, but I can say that the overall sentiment in the market has been very, very focused on the pipeline, on the assets. And I think this puts Deepcure in a great position because this has been our focus from day one. We are extremely proud of the platform we've built, but as I mentioned, it's all about the products. It's all about the drugs. And we now have a very exciting pipeline of autoimmune candidates and assets in various stages that we're developing. And I think this puts Deepcure in a great position for future fundraisers.
Kfir Schreiber, CEO and co-founder of Deepcure. Kfir, thanks so much for your time today.
Thank you very much, Danny. It was a pleasure.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.